Welcome to Neuromatter, where neuroscience becomes simple and fascinating. Today, we're diving into the building blocks of your brain, neurons and glia. These tiny powerhouses make it possible for you to think, feel, and move. Let's get started. Let's take a trip back in time to find out how we discovered neurons. Early neuroscience faced two big challenges. First was the difficulty to study tiny neurons under the compound microscope. To give you an idea, the lead tip in a pencil is about 2 millimeters across, but sizes of neurons range from about 0.01 to 0.05 millimeters across, which is 40 to 200 times smaller. Second was the difficulty to obtain very thin slices of brain tissue for viewing under the microscope. In addition, once cut, the thin slices were not resolvable until stains were developed that could selectively color different parts of cells in neural tissues. Two revolutionary stains help to see and understand neuron architecture are Golgi stain and Nissl stain. Golgi stain was developed by Camillo Golgi. This stain uses a silver chromate solution that stains the entire neuron, including its dendrites and axon, perfect for seeing neuron architecture, but only good for staining only a few neurons. Nissl stain was developed by Franz Nissl. This stain uses basic dyes such as Cresol violet, neutral red, toluidine blue, or methylene blue, to stain mostly cell bodies and nucleic acids, RNA and DNA. Good for distinguishing between neurons and glia, and for comparing neurons in different parts of the brain. Your nervous system is made up of two cell types, neurons and neuroglias. Neurons are the brain's messengers, transmitting signals. Neuroglias are the brain's support team, ensuring everything runs smoothly. Now let's zoom in on neurons, the basic functional unit of the nervous system. Dendrites are the branch-like structures receive signals from other neurons, like antennas. They contain postsynaptic membrane which houses numerous postsynaptic receptors for binding neurotransmitters. Cell body, also called soma, is the control center, where signals are processed and houses most of the organelles. Axon is the highway that carries signals to other neurons or organs. Axon hillock is the axon initial segment. Axon terminals are the end of the axon, where neurotransmitters are released to pass the message on. Myelin sheaths are insulation layers that wrap around segments of axon, allowing faster and more efficient electrical impulses transmission. Nodes of Ranvier are gaps between myelin sheaths. They contain many sodium and potassium ion channels, allowing rapid influx and efflux of ions. Now let's talk about the types of neurons. We classify neurons in several ways: by neurite shape, by dendrite shape, and by function. There are 4 neuron classifications based on neurite shape. Unipolar neurons are rare and found in the autonomic nervous system, with soma located between the two ends of the axon. Bipolar neurons are sensory cells in the retina, cochlea, and olfactory epithelium, with both an axon and a dendrite extending from the soma in opposite directions. Pseudo-unipolar neurons are common sensory neurons in dorsal root ganglions and sensory ganglia of cranial nerves, with a T-shaped process. Multipolar neurons are the most common, with many dendrites and one axon. These are your motor neurons. There are 3 neuron classifications based on dendrite shape. Stellate cells are common in cerebral cortex and cerebellum, with multiple dendrites radiating outward from the soma in various directions, giving a star-shaped. Pyramidal cells are common in cerebral cortex, with triangular soma, a single axon, apical dendrites that emerge from the pointy end of the soma, and a cluster of shorter dendrites that emerge from the rounded end, called basal dendrites. Purkinje cells are common in cerebellum, with fan-like dendritic trees that extend upward, and long, thin axons that extend downward. There are 3 neuron classifications based on function. Sensory neurons located in the peripheral nervous system detect changes and send signals to the brain. Motor neurons located in the central nervous system carry instructions from the brain to muscles. Interneurons located in the central nervous system connect sensory and motor neurons for reflexes and integration. There are 4 neuron classifications based on neurotransmitter type. Glutamatergic neurons releasing glutamate. Cholinergic neurons release acetylcholine for muscle activation. GABAergic neurons releasing GABA. Dopaminergic neurons releasing dopamine. This classification helps us understand the diversity of neurons and their specialized roles. Neurons communicate via both electrical signals and chemical signals. The electrical signals are action potentials, and the chemical signals are neurotransmitters. The action potential travels down the axon, proceeding as a wave of depolarization, results in releasing of neurotransmitters in the synaptic gap. Then again, neurotransmitters cause a rapid, temporary change in the membrane potential of the adjacent neuron to initiate an action potential in that neuron. There are a few important universal features of action potentials. Action potentials always proceed in one direction only, from the soma to the synapses, due to the refractory period of the voltage-gated ion channels. Action potentials do not vary in magnitude or speed. They are all or nothing. The parameter that can vary is the frequency of action potentials, or how many action potentials occur in a given amount of time. But neurons are not always in active state. Neurons have both resting potential and action potential. Resting potential is the membrane potential when a neuron that is not currently transmitting a signal, which is approximately -70 millivolts, maintained by the sodium-potassium pump and potassium leak channels. The sodium-potassium pump transports 3 sodium out of a cell while moving 2 potassium into the cell for each ATP molecule that is used. The potassium leak channel allows the potassium to diffuse down its concentration gradient and leak out of the cell. Action potential is a brief depolarization along the axon. It is important to note that not all depolarizations result in an action potential. The signal must cause a depolarization that reach the threshold potential, approximately -55 millivolts, for an action potential to occur. If the threshold potential is reached, voltage-gated sodium channels open quickly, and sodium rush into the cell, causing the potential to depolarize. Voltage-gated potassium channels open somewhat later than sodium channels and allow potassium to exit the cell. At the peak of an action potential, sodium channels become inactive and remain in refractory period. Potassium channels remain open as potassium continue to exit the cell, causing the potential to repolarize to its resting potential. Sodium channels reset at rest. Potassium channels are slow to close, allowing the membrane potential to transiently become more negative than resting potential or hyperpolarized. Then potassium channels completely closed, the membrane potential returned to resting potential. Neurons are not in direct physical contact with each other. There is a small gap between the two neurons called the synaptic cleft, where neurotransmitters are released by the presynaptic neuron to transmit the signal to the postsynaptic neuron. The neuron sending a signal to the next is called the presynaptic neuron, and the neuron receiving a signal is called the postsynaptic neuron. How does synaptic transmission work? Once the action potential reaches the end of the presynaptic axon bouton, the action potential depolarizes the membrane and opens voltage-gated sodium channels. Sodium ions enter the cell, further depolarizing the presynaptic membrane. This depolarization causes voltage-gated calcium channels to open in the presynaptic neuron, allowing calcium ions to enter the presynaptic neuron at the synapse. Calcium ions entering the presynaptic neuron cell initiate a signaling cascade that causes synaptic vesicles to fuse with the presynaptic membrane. The synaptic vesicles contain neurotransmitter molecules. Fusion of a vesicle with the presynaptic membrane causes neurotransmitter to be released into the synaptic cleft, the extracellular space between the presynaptic and postsynaptic membranes. The neurotransmitter diffuses across the synaptic cleft and binds to receptor proteins on the postsynaptic membrane. But neurons can't do it alone. Glial cells are their support system. In the central nervous system, we have ependymal cells, microglia, astrocytes, oligodendrocytes. In the peripheral nervous system, we find Schwann cells and satellite glial cells. Ependymal cells line the ventricles and the central canal, assist in producing, circulating, and monitoring of cerebrospinal fluid. Microglia are the cleanup crews, removing debris, wastes, and pathogens by phagocytosis, fight off infections, and protect neurons from damage. Astrocytes are the brain's caretakers, maintaining blood-brain barrier, providing structural support, regulating ion and nutrient, dissolving gas concentrations, absorbing and recycling neurotransmitters, and forming scar tissue after injury. Oligodendrocytes responsible for myelination in the central nervous system. In the peripheral nervous system, we find Schwann cells responsible for myelination in the peripheral nervous system and participate in repair process after injury. Satellite glial cells surround sensory neuron soma and regulating oxygen, carbon dioxide, nutrient, and neurotransmitter levels around neurons in ganglia. Why myelin matters? Myelin acts as insulation, speeding up signal transmission in axons. In the CNS, oligodendrocytes handle myelination. In the PNS, Schwann cells take the lead. However, keep in mind that all axons have some degree of myelin wrapped around them. Some are classified as being unmyelinated because the degree of myelination is very low when compared to myelinated axons. Even unmyelinated fibers get some protection from Schwann cells in structures called Remak bundles. The more myelination, the faster the signal. If you enjoyed this dive into Neuron 101, don't forget to like, subscribe,